Okay, so welcome everyone to this program. What we're going to be doing in this program is going over the very basics. And so I want to start by briefly introducing bioinformatics and kind of asking you to, reply some of the questions so that we can together think about why bioinformatics and what is it useful for, why should we complete this course? So first of all, bioinformatics is this field that tends to attract people with computer science background, maybe with biology background. And the whole purpose of this field is to develop tools and approaches to analyze data that comes from the biological domain. And so I think the obvious question that rises up is, why is bioinformatics actually needed? So let's go through some of these reasons together. And if you have any additional reasons that you think, okay, sorry about that. So let us change this to here. There we go. So why are we, interested in bioinformatics, and why is bioinformatics actually needed? So number one, lots of data is being generated, right? So the availability of data, just the volume of data, all of these kind of changes in the biological domain lead to the fact that we have to have some method of structuring this data, organizing this data, making sense of it. What is it actually that we are finding? When people generate data, for example, biologists, obviously understanding what are the important things in that data is going to be very important for you as a biologist, because in many cases, biologists used to just give this data over to somebody else. Someone else needs to look at the data and try to infer from this data, what are all of the issues that could be coming with the actual signal that you are trying to study. When we rely on publications refer to methods of analysis, annotation, and interpretation of biological data. Because this is a highly dynamic field, methods are often updated, reference databases are often updated, and also new understanding of what are some of the basic technical challenges with different types of data. All of these issues have to lead us to being up to date and kind of understanding the language of bioinformatics. Many assumptions are, when you think of a hypothesis, right? When you think of a new project, a new idea that you want to test and you want to generate some data, before generating data, which is expensive, it's very useful to know that there are many databases where you can check your assumptions and see whether somebody else already tried that kind of an experiment, and not only look at the findings that they report in a publication, but also look at their raw data. So we can find other types of experimental results, and we can try to infer something from those. Collaborations, right? In many cases, working together with others requires you to have a good method of communication. And when somebody produces some kind of p-value or some kind of number, you want to know what that number means, but also where did it come from? And because people come from many different backgrounds and approach a data set from different perspectives, if you are the one that understands the biology of that data set, you should be equipped to be looking and interrogating into that data set with an understanding. And number 6, we see that data contains noise and artifacts. This is an important point because in so many cases, the assumptions about data and what the data represents, if we just follow standard methods, can be misleading and can actually be even bad because we're not taking into account all kinds of technical variations. So knowing how to address the technical issues and know what the issues might be are all a part of bioinformatics. Many different labs perform experiments to annotate biological phenomena. So because this is a team effort, many different people are involved in similar domains. Knowing what else is being available and how to interpret that is important. And then finally, there are many applications of data analysis. So once that data is generated, many domains, including basic research and translational research, are basing whatever they're doing on that type of data. So healthcare, pharmaceutical industry, agriculture, ecological research, biotechnology industries, all of these rely on this data. And so knowing how to generate, knowing how to annotate, integrate, and analyze this data is very critical. So Bioinformatics, bioinformatics is essentially the intersection of information technologies from the informatics side or computer sciences, statistics with analytical methods, and the application. That application, we can kind of group it to be about biology, but in reality, there are many different aspects of application of bioinformatics. And so as we will see in some of these introductory courses, the applications of bioinformatics in part is what makes it exciting. And in part, it is also challenging because of the quantity and the quality of data that these applications that are very ambitious require. So when we are going to be talking about all of these aspects in this particular course, a lot of the examples that we're going to be using come from the biomedical domain. So even though there are many applications in agriculture and in maybe biotechnology and other fields or ecology, we're going to primarily be looking at different types of applications in the biomedical domain. And in the biomedical domain, we're assuming that the translation of research comes from a basic understanding of some biological phenomena that could be used in clinical practice ultimately. It might not yet be used in clinical practice, it might not ever be used in clinical practice, but it is going into that direction. And so what's important is that not only we need to understand the actual biology at the level of individual elements that have to interact and work together and understanding those mechanisms, we also need to think about the application downstream. So what we're going to be doing in this program is taking a look at how big data, this is called big data bioinformatics, can be applied in some aspect of, let's say, precision medicine. So what that means is that we're going to be looking at some aspects of the diagnostic of the diagnosis of disease, how to detect and determine what a disease is, and the application of this kind of information to intervention. And typically we think of that in terms of therapeutics. So the process of annotating and interrogating available data leads us to understand how this data describes a biological process that typically leads to disease or accelerates kind of disease. And as a result, we can identify interventions that are either new, like drug discovery, or repurposed, or maybe more precise, or maybe more defined. And so those are some of the issues that we're going to be looking at within the scope of this introductory program. So the introduction will deal with the intersection of biology and data. And we will also speak about what are some of the technologies that allow us to even think about the way that deep biological understanding of biological processes and biological phenomena, how can that be translated into the clinic? One of the ways that we can do this today is because of this accelerating pace of technological innovation. And because of technological innovation, we now can very quickly identify what is going on at the biological level and translate that to some kind of an application. So there are two key technologies that we're going to be speaking about: the computational technology, or ability to process and analyze large quantities of data, and sequencing technology as an example of one technological innovations that has transformed our understanding of omics data. So before we move on, let's briefly define what omics data is. Omics data represents many different types or collections of different types of data. So for example, we can say phenomics or genomics, epigenomics, transcriptomics, metagenomics. When we say those words, we're referring to collections of data, and those collections of data represent some very specific things. So for example, when we say phenomics, we typically can mean some kind of a visual, maybe morphological characterization of data. Typically, omics refers to high throughput automated technologies that allow us to generate large quantities of data. So for example, for phenotyping, we can use imaging, and then analysis of that imaging data can help us understand differences between cell morphology, or maybe tissue specificity, maybe identification of tumors and things like that. Genomics refers to the genome. What are the changes in the DNA at the nucleotide, amino acid level, or structural reorganization level, such as chromosome rearrangements, copy number variation, etc. Epigenetics refers to the study of epigenetic regulation of gene expression. So we can look at epigenomic data like chip sequencing, bisulfite sequencing, to study DNA methylation and chromatin immunoprecipitation. And transcriptomics refers to the gene expression, the collection of expressed genes and transcripts or isoforms of those genes. And ultimately, we can also expand this to proteomics, metabolomics, etc. So there are many different types of omics data. In this program, we are going to primarily speak about genomics and transcriptomics and a little bit about metagenomics, but we are going to do it at a very superficial level. The reason for this is because we want to provide a broad overview of how different types of omics data could be generated with the same technology, sequencing, and how studying those sequencing data sets, we can find relevant and applicable biomedically important phenomena that we can study. Where can we find such data sets? So NCBI, the National Center for Biotechnology Information, is a major resource of freely available data sets. We know that this data set is not only rapidly accumulating more and more data, it is also becoming more and more easily to use. So previously you might have participated in some kind of a workshop or heard about a workshop on just how to use NCBI, how to find data on NCBI, how to maybe analyze some of the data using NCBI tools. And we're going to briefly talk about that in this program as well. But we will also see that actually the website becomes more and more intuitive. The data set are better annotated. There are more of them. It's easier to find them. And so once you get a hang of what's in there, you will be able to start thinking about what can I do with this? So it's an important resource. Again, over the years, from about the 1990s up until today, there are over 100,000, we need to update this slide, but there are over 100,000 publicly available data sets just on one repository called GEO, Gene Expression Omnibus. So Gene Expression Omnibus would, of course, have collections of data from transcriptomics. We will see how this data is easy to download or even analyze without downloading to your computer. But definitely you will also find this an important collection of different types of other data, including metadata about what the project was about, what kinds of samples are available, etc. So this program, Introduction to Bioinformatics, is going to deal with the following topics. We will talk about bioinformatics in general, what are the emerging technologies, what are some of the important concepts, and what are the applications of bioinformatics. We will then take a look at some of these specific types of technologies. DNA variations and mutations, what are they? How do we find them? What do they mean? How do we interpret them? Transcriptomics, what are genes? What are transcripts? How can we measure expression of those genes? And how can we annotate them to understand how are they associated with some kind of phenotype? We will also look at some applications of methods. So we will look at statistical methods of analysis as well as machine learning. So why machine learning and how do we use machine learning specifically for biomedical or biological data? You will be introduced to some tools to do all of these, including, I will show you in a few minutes, some of the tools that we have for easy, user-friendly analysis of big data, but also some elements of coding, because it's important to understand how a bioinformatician might deal with data, maybe what are some more efficient ways to start analyzing your own data, or maybe even just looking at data sets that you already have, how to open them, how to get started in that. And then we will look at some of the projects. How do you actually take what you have learned and apply it to different examples of data sets taken from cancer, infectious diseases, and biotechnology domains? And that will give you a glimpse of bioinformatics as a kind of integral component of many of these different fields. So this is an overview of the program, because the program is going to be primarily online and self-guided, we're going to rely on the Omics Logic portal. So as Farhana already mentioned, the Omics Logic portal contains self-guided asynchronous materials that you can use, but also provides you with a few other resources that I want to go over. Before we go into that, does anyone have any questions about bioinformatics in general, or what we are going to learn in this program, or maybe how long the program is, or how to participate in the Q&A sessions? Does anyone have any questions about that? Or maybe about registration, how to register for the program if you haven't completed your registration? Okay, so if there are no other questions, I will just repeat again for everyone. This is the portal, learnomixlogic.com. Okay, we do have a question. Is there an exam at the end of the course? Yes, so I will address that in a moment. Okay, so let me show you then how this course will practically work. So again, the objective of the course is to provide you with the resources and the materials that you can complete on your own time. But behind the scenes, we're going to be tracking everybody's activity to make sure that you have completed the minimal number of lessons and have completed the exercises or quizzes that are provided within these courses. And if you have any questions, we're here to help. We're here to support your journey, and we're hoping to help you complete this successfully and be able to apply these skills in some meaningful way. Will this course enable us to not only analyze our own data, but also expand to analyzing many data sets? Great question. Yes, so we are going to be using primarily other people's data sets. We're not going to be using your data sets in the context of the training. But after you have completed your training, you will be able to analyze some of your own data or use other people's data, like I said, for example, from NCBI, to really understand what's going on in terms of other people's experiments. Okay, so as Farhana already mentioned, when you get to the portal, right here on the very top, you will find your email address. So let me ask you here, if you already have registered, you have an account, please put number 1 in the chat box so that I know how many of you have already created an account. Excellent, thank you. All right, perfect. So if you still do not have an account, it's free. You don't need anything to create an account. And later on, once you have created your account, we will guide you on how to complete the program registration. So once you're here, if you click on this, you will see not only, like Farhana showed, the profile information, but down here, you will find a number. Well, for you, probably you will just have one program. And so one of these programs that you will be a part of is called, is the program that we are in now. And this program is called Biomed Introduction to Bioinformatics. Biomed stands for the Department of, Farhana can say exactly what, Biotechnology and Molecular Medicine, I believe, at LSU, at the vet school at LSU.
Yeah, you're right.
Okay, great. So this program is going to give you an overview of everything that you need to kind of go through this. For example, you will find here the courses that you have to go through, and you will also see what your certificate is going to look like at the end. So once you complete all of the sessions and earn the points associated with these sessions, you will be granted this certificate here online. But I will go through some of the other certificates that you will find inside every course. You will also down at the bottom find a link to a Slack channel. Now, a lot of people get confused with the Slack channel. Do I have to join Slack? Do I have to register for Slack? Basically, every channel you have to register, and once you're registered, then you will be able to access the discussion. So here you can ask a question, and for example, well, let's do it like this. Biomed Introduction to Bioinformatics. So on Slack, if you have any questions, if you have any technical issues, you will be able to find it on Slack and ask someone about those questions. Okay, so this is the Slack channel. And if you do a question like this, for example, okay, so perfect. If you have any, if you have issues accessing the Slack channel, you will be, please email this email. And we can help you. Okay, and so either this one or Farhana, if you can add your email there as well. So if you have any questions, we'll guide you on how to do that. Now, why would you need access to the Slack channel? The Slack channel is a good way to get specific answers to your questions. For example, you're trying to access the course and something is not working. You're trying to submit assignments and you don't know who to send them to. You're struggling with some piece of code and that code works on our portal but doesn't work on your system, right? This is a place where you can directly ask those kinds of questions. And if you have, maybe others can suggest that they know the answer, or our team will actually take a look at it within 24 hours. So with this program, you will see here the overview right here on top, the courses, and then also Q&A at the bottom and the certificate. So we went over those things. As you will see on your profile, this will turn into activity over time. So the activity over time will result in completed courses. So those courses, while you're in this program for the next 3 months, you're able to take any of the courses on this portal, all of the projects, all of the courses, even the more advanced ones. And you don't have to worry about paying for anything, because if you're a part of this program, everything's already paid for. So you have access to our support team, you have access to the coursework. If you want to try more advanced courses while you're in this program, you're welcome to do so. Another important resource that you will have available to you is the big data analysis platform called the Tbieinfo platform. How many of you have already tried accessing this platform, either by taking one of the courses or just by seeing that in the description? Did anyone try to log in and see if this is accessible to you? Yes, excellent. Okay, so if you have registered for the Learn portal within 24 hours, you should be granted access to this portal as well. If you don't, and how do I know that I have access? Right here, if I hover over this icon, I will see my email address here as well. So if you don't have access, please reach out to us. Again, the same email, support@PineBio, and Farhana, if you can add your email there as well, that would be useful. So once you are in here, this will be useful for several reasons. First of all, you will be able to understand how different types of data is analyzed by seeing examples. So the examples are accessible by clicking on this drop-down arrow. Okay, once you click on the drop-down arrow, you will find a demo pipeline. This demo pipeline will show you how individual scripts or analysis algorithms are connected into pipelines or workflows. And as you go through the course, you will learn about each one of the steps that you're taking in this pipeline to understand what it means and why to use this one or an alternative, and how specifically to use it. So for example, when we speak about transcriptomic data, you will be learning about the basic steps of transcriptomic data. Then you will be shown some limited number of options that you can try. After you try those options, you will have some results, and you can always go back by clicking with your right mouse button and change your selection. So this is something that you will use to understand different types of omics data and eventually different types of applications of analysis. Okay, so in this, also in this section, you will find short descriptions for different types of sections that we have here. And right here under tutorials, if you just click on tutorials, if you need a quick reference how to use a specific method, you can go in here and find a much shorter version of a brief introduction to a section. So if you ever have any questions, you can see here, for example, how to upload files or how to run a particular type of an analysis. So this is a useful resource here as well. Okay, so the resources again include access to the Learn portal. On the Learn portal, okay, Gabella is asking, how do we gain access to this? Do we have to make an account here as well? No, you just log in. Yeah, if you were already signed up and you had access to the Learn portal, all you need to do is you need to go to this portal and sign in using the same credentials. If you don't have access, your credentials are not working, please email us. So you can email again Farhana or support@pine.bio. Okay, so we will try to address all of the sign up issues and access issues before the end of the week, as the program just started. Does anyone else have issues accessing the portal? So I know that Gabella, okay, Chris, thank you. So if you could just say that you can't access so that we can write your name down and then we can reach out to you when we've given you access. Perfect, yeah, so everyone, if you would like to. Okay, perfect. Thank you, Tessia. I think that some of you just registered today, so it will take a little bit of time. You know, after you have registered, again, for those of you that are from LSU, you should have completed your registration through the iLab portal. For those of you that are from NAIPI, you basically gain access by filling out a form. We should have emailed you. If you just signed up today, then you should get the email tomorrow. And if you don't have access and you're not from either NAIPI, LBRN, or LSU, then please reach out to us again, support or Farhana, and we will help you with that as well. Perfect, thank you, Cabela, and everyone else. Okay, so again, how can you organize your time to best make use of these resources in the 3 months that we will have? The first section that we have designed is a section called Getting Started. So what does Getting Started include? It includes 3 courses. One of them is Introduction to Bioinformatics that primarily talks about, you know, very high level overview of what is bioinformatics, how's it used, different examples of how it's used in many different exciting fields, etc. But it's really an introductory course. Now, in this course, the first lesson is going to be a survey. This survey is to better understand your background and to better help suggest the best course of action for you as you embark on this journey to learn bioinformatics. The second course is called Bytes and Molecules. Here you will find terminology about different things that we will deal with. For example, what are genes and how are they different from isoforms? What's the difference between single nucleotide variant and single nucleotide polymorphism? What is a mutation? How does a change in sequence affect the structure of a protein? So all of these basic concepts are going to be relied upon in the coursework that you will go through. So if you don't know a lot of these terms, you should take a look at that course first. Also, you will find there specific applications of bioinformatics in the domains of precision medicine, space omics, and other exciting fields. And we'll have a very short introduction to coding. And then for those of you that are interested in continuing and expand your technical skills, we have an introduction to bioinformatics in R and introduction to bioinformatics in Python. So I want to spend just a couple of minutes explaining what are these courses and how to use them. So again, if you go to the courses tab and you scroll down here to getting started, this will take you to the collection that I was just explaining. You'll have here bioinformatics in Python or bioinformatics in R. You will also find a very brief introduction to coding right here in bytes and molecules, right here, bioinfo in R. Okay, so what can you learn in a course like this? First of all, R is a very useful way to get started with coding, especially if you're a biologist and you don't know any coding. It's very simple, it is free, and you will be able to do a lot of things, including images and figures that you will see in many publications. They were produced in R. So this is a good way to get started and quickly get to where you want to be in terms of publications if you're a graduate student or if you're planning to publish your research. So if you click in each lesson, you will have some kind of a description, then you click on next, and you will be given a very brief piece of code. So you can see that the actual code here is one line, and this one line describes how to read the table. So this is a major problem for everyone who's getting started with coding. How do I not just write hello world, but how do I actually load my own data so I can analyze something that I understand? And so here you will find some data objects. How do you actually deal with data in a coding environment? And then you will be given several examples. So what can you do with this kind of data? Here we take an example from a gene expression table. And so we will look at different examples of how to make some plots. And that will give you a good sense of what is expected. Then there's going to be a practical application. So not only do you load the data, right, theoretically, but you can run it right here in your browser. So you don't have to do anything. If you're just learning how to do R, you don't have to do anything, install anything, nothing like that. And everything that you do is going to be using some links to data that we uploaded to GitHub. So by doing this, you're already learning how to load data and how to manage your data on the cloud, let's say. So the first exercise is just to learn syntax. You can edit what's here. And as you read the instructions, this is very important. A lot of people try to do this without reading instructions. Read the instructions. What it says is to use this, you have to copy the link. So I right click on this and I copy the link, and then I replace this part of the code with this link. If I have done this correctly, the code is going to load the data into a variable called df, and then I'm going to check the dimensions of df. So I can just click here on run. Okay, and you can see that success running code, but answer is incorrect. Okay, so something worked, the data is here, but it is incorrect. So if you don't know why it's incorrect, you need to go back here and you need to understand the syntax, right? So what is the syntax? The syntax is how to write commands that the computer will understand, because every language has an interpreter, sorry. So here you would want to understand what are these things and how to use this to do what the assignment tells you. So let's take a look at what does the assignment says. Okay, the assignment says check dimensions. So to check dimensions, you have to write dim. You can clear your code console like this, run it again, and now the code is correct. Okay, so this is a basic way to use this console inside your browser. If you are going to be learning how to code and you're trying to understand how to code, to visualize data, to analyze data, etc., this is going to be very useful. So why the big data platform and coding? The answer to that is simply because we are going to be looking at large data sets. Okay, so I will go again in here, and I want to briefly explain the difference between these tools. A lot of the standard visualization tools are going to be available here as well. You will find here different utilities, so you can perform normalization or run blast or make a heat map and things like that. But these are downstream things, right? These are kind of at the final stages of your analysis. Typically, one of the biggest challenge is that the data sets are huge. You need to analyze them, and you don't have computational power on your own computer, your laptop, to be able to do that. So processing data or turning raw large data sets into manageable tables or data sets that are structured, you can use a platform like this. Just to give you a sense of the size of this data, if you go into the first section and you click on start, you will see right here the input files. And if you look, what are these files? And what are the output files, right? You will be able to see that each step of the way creates a number of files that take in whatever you give it, then produce a file that is the result, pass that result to the next step, etc. And this pipeline ultimately will generate gigabytes and gigabytes of data. So it is not very effective to work with data sets that have more than several samples per group. So maybe between 10 and 20 samples is a typical sample size today in an experiment, maybe now more and more towards hundreds of samples per condition. So those are just not manageable on your local infrastructure. And so what we will do is we will process the data using tools that are already here. We will take the results that are structured and then deal with those either again in an interface like this that is user-friendly, or learn how to produce plots on Excel or in R or in Python. Does anyone have any questions about this or how to use the coding elements in these tutorials? Okay, if you think of any questions, don't hesitate to let me know. Now, if you briefly go back to your profile, you will see that all of this activity is going to be added up. So for example, you can see that for each step, you will get a number of points. And the number of points accumulates right here on this left side. And your goal within this course is to get to 10,000 points. These 10,000 points are going to go towards your certificate. So if you want to obtain a certificate from either LBRN, LSU, or NAIPI, you will have to get 10,000 points. How do you get 10,000 points? You get points for individual activities, you get points for completing lessons, courses, and attending sessions. These sessions are going to be either live or recorded. Now, if they are recorded, you should be able to find them right here. Okay, so this is it. Yeah. If they're recorded, you will be able to find them right here. For example, you can see that this is today's session. Today's session, Introduction to Big Data in Biology. After today's session, these are some associated online resources that you can start with. For example, Introduction to Bioinformatics course, access to the educational portal, access to the analytical server. All of these things are something that you want to take a look at. For example, you can click right here and get to the Introduction to Bioinformatics course. Or after you have completed this, click on Complete Session. Okay, so once you complete the session, you see that session completed is green. Again, you can go to your profile and you will see that you get 10 points for completed program sessions. Now, these are going to be critical towards completing the program. So completing the program means that you will have completed all of the sessions that are found on this program page. Okay, so again, this is the page. You should be able to see on this page what I see. which is my profile, which you can expand and kind of see what your current stats are. And then you should be able to see sessions that you can complete. Okay? If you don't see sessions that you can complete, and here you see pricing information, that means that you have not been assigned to this program as of yet. And if you haven't, that means that we just have to go through the steps of the registration. And there are several different steps for registration for people from LSU, Louisiana Biomedical Research Network, NAIPI, or outside of these organizations. Okay, so after you have gone through this series of courses, what you should be able to understand is the role of bioinformatics in application to different challenges in biomedicine, in basic and translational research, as well as in industry like healthcare and pharmaceutical and biotechnology industries. You should also be able to speak bioinformatics. So you should be able to converse with somebody who's knowledgeable in bioinformatics, maybe discuss with them methods, maybe images or specific figures that you want for your publication. You should be able to read through the methods section of any paper and more or less understand what was being done. So that terminology and the logic behind of the analyses that you typically use in these biomedical research papers should be more or less clear. You should also be familiar with the very basics of coding. Not only understanding how to piece together an analysis process, but for specific stages of that analysis, how would you be able to do that on your own? Now, that could be done in Excel, in R, in Python, or on the platform. So you will be introduced to a variety of tools. After this, we have regular sessions on different types of data. And so for each type of the data, like genomics, metagenomics, and others, you will have to go a little bit deeper to understand how to practically deal with those kinds of data sets and see some examples. So that's why we have additional 2 programs that are going on right now, genomics and metagenomics. Those programs are available to anyone who already has a little bit of background. Do we need to start an application to access the class in the link above? Well, so that link is for those that are not a part of NAIPI, LSU, or LBRN, Sebastian. So if you are a part of LSU, LBRN, or NAIPI, then there are different ways to get access. So if you type in chat, what, are you, if you are a part or not, I can give you better directions after that. Okay, if you're a part of LSU, then iLab is the way to go. So Farhana can help you with iLab, how to get registered there. So in parallel to this program, Introduction to Bioinformatics, we have these additional ones, genomics and metagenomics. Those are specific to different types of data. And then we also have a program called data science. So if you already know the basics, you kind of speak bioinformatics, you know what the general idea is, you would like to get more into the advanced methods of data visualization, machine learning, deep learning, that is the program for you, data science. You will be able to learn how to code more effectively in R and Python, and you will be able to apply more advanced methods of analysis and extract meaningful insights from larger data sets using coding. So that is a program that is also starting this week. All of these programs are starting simultaneously. If you're just beginning and you don't feel comfortable doing this just yet, then you should complete this program first, and then you will be eligible to participate in the other programs as well. Now, after these programs, which are basic programs, they speak about the basics of data types or skills. We then have a series of specializations. These specializations are designed to introduce you to projects sourced from publications, where we take those examples from publications and we start speaking about the methods in the context of a particular question, a research question. And so that is the project component of the Omics Logic portal that allows you to not only apply bioinformatics technically, but use it as a way to conduct research. asking questions, developing hypotheses, and translating your understanding of data to a step-by-step process that will give you the result that you are looking. So those divide up into oncology, infectious diseases, neuroscience, and agriculture. Specifically, if you're interested in any of those, we have infectious diseases, bioinformatics for infectious diseases starting soon. If you're interested in that, you're welcome to write me an email, for example, or you will probably receive a notice about that as well. But if you have specific questions about that program that's starting very soon, you can reach out to me directly. So as I mentioned, these are all possible to analyze using the Tbioinfo platform. So if you don't want to do coding, you really just want to understand how these different elements connect together and how to get the figure that you need, or how to process data and get the table that you need. This is a platform that will suit you the best, and you can complete everything that we discussed today on this platform itself, including downloading codes to perform specific types of visualization and statistical analysis and machine learning. So if you just want to get those codes and you want to understand the logic behind the analysis, just go through the steps and then you will be able to download the code. So to summarize, Bioinformatics, or Introduction to Bioinformatics, is a program that is designed to provide you with a high-level overview of some of the basic things that you need to know to get started with bioinformatics, and give you access to resources that will support your individual path from knowing basically nothing about bioinformatics to being knowledgeable and more or less proficient to do some of the basics. And after that, you will be able to apply your understanding to different areas. Either you want to become technically sound and proficient at bioinformatics, you want to understand the concept of bioinformatics and the language of bioinformatics to communicate better with others and read publications, or you just want to know what bioinformatics is, how is data used in a variety of domains, and how should you prepare for that if you're, for example, an undergraduate student, and maybe that will have an impact on your career as a biologist or a clinician. With that, I am going to stop sharing my screen. And I do want to ask if you have any final questions before we close this session. Again, the sessions are going to be available both live. Some of the sessions are going to be live. We will have some Q&A sessions. And we will have asynchronous sessions that are going to follow some of the things that we discuss. So 2 dates that are important to remember. One date is February 11, and another date is February 18. Those are going to be the Q&A sessions. Okay, so I'm going to put this in chat. February 11 and February 18. So these are Q&A sessions. If you're a part of this program, we highly recommend that not only you participate, but also allow us to prepare the answers to your questions. So email us or post it on Slack, and we will be able to answer your questions in that session. So typically people struggle with some practical hands-on things. You definitely want to be able to prepare us to address those questions specifically. And so you can email us ahead of time so that our team can prepare for those. Again, those that have registered already, you will have access to everything that we discussed today, starting today, and all the way up until the end of March. If you have any questions about access or registration, please email Farhana, fmusar1@lsu.edu. She's the coordinator for everyone that is from LSU, Louisiana State University, and LBRN, Louisiana Biomedical Research Network. For anyone that has questions about access through NAIPI, the National Association of Idea Principal Investigators. The NAIPI has provided us with 50 licenses for faculty and students that come from institutions funded by the IDEA program. So if your institution has a COBRI or an INBRI and has funding from the IDEA states, you will have a PI who is a part of NAIPI. If you have such a person, typically this will be a faculty member, you can find them on the NAIPI website. The NAIPI website is NAIPI.org. I will paste that link here as well. If you don't know if you're a part of NAIPI, go on this website, you will see there a map. And on this map, you can click on the map and you will see the list of all PIs. If you have that PI's email address or something like that, reach out to them. We will need that information from you for registration. If you are outside of LSU, LBRN, or NAIPI, you are welcome to proceed with the registration that was on the page that I already shared. So the page, the program page, gives you some, gives you ways to access the program directly without funding from an institution. So you can either pursue this direct link, or if you are a student and you would like to get a scholarship, please reach out to us as well. So the best way is to reach out to marketing@pine.bio or support@pine.bio. Okay, how do you check if someone from our university is or is not from NAIPI? So again, the best way to do this is to go to the NAIPI website. Hold on, let me switch the screen. Okay, so if you go to NAIPI.org, so this is the link. You scroll down and you will see here a map. So if you know which state you're in, you should be able to click on that state. For example, I am in Louisiana. And here you will see who, if you scroll down, this will have all of your NAIPI PIs. So all of these PIs are members of NAIPI. So if you see them, they will be either a COBRI PI or an INBRI PI or a CTR. If you see someone from your university that is here, that's enough, right? You just need to mention who that is and which university that is. And then the NAIPI organization will provide you with funding for these programs. All right. Any other questions? Have the links for the genomics, metagenomics orientation been sent out, and when are those? I do believe that they have been sent out. So again, let me check my calendar just to see. I do see that we have a session on Wednesday for genomics and a session on Friday for data science. So if you're interested in those, I believe if you have registered somewhere, you should be getting an email from Somalika Ray or Farhana if you're from LSU or LBRN. If you didn't get those, then you're welcome to email me as well, and I will be able to send that information to you. But typically, these sessions are going to be at this time from 3 to 4 p.m. First, we were going to do it at 1, but then some issues with scheduling came up. So now it's at 3 p.m., 3 to 4 p.m. They will be recorded. So if you can't participate, that's fine. We will send you a recording by link if you are part of the programs. Are there any other questions that I can answer today? Yes, 3 p.m. Central Time. When should we expect the emails for the Zoom links for the data science course? Again, that should be, I think the session is on Friday. So if you don't receive it by maybe Wednesday, please reach out to us. Because of so many different ways to register, sometimes we miss out on people's information. And so if you don't get those, please do reach out to us and we will be able to share that with you directly. Okay, well, we're a little bit past our time, so I will let everyone go. Thank you for joining today. If you do have any other questions, you're free to email me. If you have other people that you think are a good fit for this training, please share this information with them as well. And I will be looking forward to seeing you again in our next Q&A session. Thank you.